So my name is Heather Gregg, and I'm a second year's master's pathway student. And so I'm in the Anuj Kumar lab, which is in molecular, cellular, and developmental biology. And then I'm also Native American, so I specifically come from the Chippewa tribe. I'm actually found in the Bear Clan up in the Upper Peninsula. Originally, when I was in high school, I actually didn't even want to go to college. I was like, this isn't for me. I'm not doing this. But my parents forced me and told me, you have to do 1 year, because as a Native American in Michigan, we get the Michigan tuition waivers. So they're like, you're at least doing 1 year, whether you like it or not. So I was like, okay, fine, I'll do 1 year and then I'm going to quit. Well, I started off and I was like, well, I like science a little bit. So I went ahead and started as a pre-med major. Well, the first year, I really liked the stuff that we were learning. So I was like, well, hey, maybe I want to be a doctor. And so I started job shadowing my sophomore year. And I didn't really like the experience at all, but I was like, well, there isn't really anything else to do. And plus, I want to be able to provide for my family. And so I just kept on job shadowing with various different doctors. And then my junior year at our school, we were forced to do a senior thesis project. And so I actually started doing some research on the side, even though we didn't have actual labs like in the University of Michigan. We just did on the side work in the classrooms. And so I did my senior thesis project, and I also did a few other research projects. And I was like, this stuff is actually kind of interesting. I feel like I could do this. But I was like, well, I've already committed to being a doctor. I gotta, be a doctor. Well, then my senior year comes, and I was like, and I rotate with this one doctor. I was like, I can't do this anymore. I can't keep on faking and acting like I want to do this when I don't want to do this. And so then my senior year, I switched to research, and I applied to a bunch of different master's programs from various different schools. And that's how I came to the University of Michigan, because they have this master's pathways program. And so this program specifically is a program designed to help underrepresented students get their hands into a real research lab, as well as you get teaching experience, and then you also, do classes like a student. So it's a really busy program, but I really enjoy it. And so that's actually how I came to be here. And plus, you actually get the experience of a PhD student, because you get a stipend instead of having to pay. So that's also a really cool thing too. And so even though I was doing research here, I realized also that I really love mentoring and teaching students. And so that's kind of where I'm gearing my professional career to. My biggest struggle is that in undergrad, I was always someone that thought they had all the right answers and they knew how to do everything. So when I came here, I realized that I didn't even understand basic techniques. And so that was really hard for me to get over. And it really caused me to struggle with a lot of mental health issues. Work-life balance is a huge struggle, because I know for my husband and I don't know if anybody else has this same problem, but we work completely opposite schedules. So I'm typically in the lab from 8 to 7 every day, and then I come home and I'm studying. But my husband is at work until midnight, but I have to go to bed by like 11:30. So we really only get weekends off together, because my particular lab doesn't make me work on the weekends. I just have to work Monday through Friday. And so we only have Saturday and Sunday together. And so we have to really, be purposeful with our time. And I really have to make sure that I have everything done the weekdays, so then weekend we can just be together. So I typically do at least 10 hours a day, 3 days a week. And the reason why I do that is because I specifically use Tuesdays and Thursdays to work on my homework and focus my time with my students. So then Monday, Wednesday, Friday, I can just be straight lab and not think about anything else, just focus on my lab and my research work. So that's just personally how I do it, and that's still the way it's worked for me. I really like microbiology in general, so anything that has to do with microbiology is very fascinating to me. I know when I did an undergrad is I specifically studied microbiome work, which I'd like to go back for my PhD to do stuff like that again. but I do really like understanding how all these molecular pathways actually cause changes to the phenotype, which is what makes all these different pathogens so virulent. So it's not like they just wake up one day and they want to infect everything. It's actually because of these specific genes that are being turned on and expressed. So that was kind of really cool because I was never really into molecular biology until I came here. So it's kind of cool how you get forced into things. Gosh. Okay, so COVID literally shut down my research for 4 months. And so I couldn't do anything, which means I will not be able to get out a paper this year, which is very sad. I know my mentor and I are talking about having at least all the data ready. So then next year I can help him write the paper. So then we can at least have a publication by 2022, we're thinking. But so it's been a real hindrance. And then on top of that, there's all these rules we have to follow. And so that's a little hard for me to make sure that I can get scope time because you have to schedule with everybody. So I can only get on scope like once a week. So I have to be really purposeful with my time there too. So it's been just a little stressful because a lot of my stuff I can't do at home. Like I haven't gotten enough data to do that yet. So I still have to do a lot of lab work. And so the specific project that I'm working on is understanding the role of flow 8 regulation of phospholipase C in pseudohyphal growth in Saccharomyces cerevisiae. And that probably sounds like a bunch of gibberish. But just some general info about our model organism that we use. And so we actually use this and then Candida albicans as well. And so this is a specific type of yeast and fungi. And so the reason why we look at this is because there's a haploid form and a diploid form of this yeast, and it's really easy to see genetic mutations, see genetic differences, and just see phenotypic differences when we mutate these genes. And so it's a really good way just to understand genetic engineering and how we can change that to what we want to visualize. And so specifically our lab focuses on pseudohyphal growth in this species. And so pseudohyphal growth, since a lot of people don't know what this is when the tissue becomes invasive. And so this is due to nutrient starvation. So we specifically focus on pseudohyphal growth. And the reason why that's really important is because this is what causes virulence and opportunistic pathogens such as like Candida albicans and other yeasts. And so if we understand how this mechanism works and how these different genes affect this type of growth, then we can understand how to create antifungal drugs in order to selectively only affect yeast as opposed to humans. And so then my project specifically comes off this study. It was actually done by a former grad student in the Anuj Kumar lab. And so she was specifically looking at inositol polyphosphates, which were then used to regulate and predict pseudohyphal growth. And so what she did is she deleted and overexpressed specific kinases, which are proteins that phosphorylate different genes in order to identify elevated levels of inositol polyphosphates. And so when these deletions occurred, or overexpressed, then filamentous growth was then shown. And so then this showed, okay, and this specific pathway that we're looking at, because we look at 4 different pathways, this then demonstrated that, okay, when we overexpress this specific gene, which we looked at KSS1 and FUSE3, then we see an overabundance of pseudohyphal growth. So this must be a gene that we should probably target. And so then that then led me to another specific transcription factor that was found in this paper known as FLO8. And that's actually what I'm specifically looking at, is seeing how the expression of FLO8 then induces pseudohyphal growth. And so the purpose of my study is to analyze whether or not FLO8, the transcription factor, activates phospholipase C, which is a specific lipase found in the inositol polyphosphate pathway that produces a specific inositol polyphosphate, which is known and that's tau polyphosphate 3, which is a really well-known study in this area of research. And so then I'm basically trying to see whether or not this actually activates the transcription of PLC1. And so the way that I'm planning on doing this is creating deletion mutants of flow 8. Sorry, flow 11 is a whole nother transcription factor. And I actually did successfully create these deletion mutants. And so I have a flow 8 deletion mutant, and then I also have an MSS11 deletion mutant, because this is our backup if flow 8 isn't actually increasing or decreasing phospholipase activity. And so then we're going to extract the RNA and then measure the RNA levels under nitrogen conditions and seeing whether or not phospholipase C activity increases or decreases. And then we're going to use chip analysis, which is a specific type of protein analysis that will tell us whether or not where flow 8 binds, if it binds at the promoter region or at a different region found on the DNA. And so then if flow 8 doesn't cause any changes in RNA sequencing, then we're going to try MSS11 as our backup. And so yeah, that's my project.